Okay, students. So now we are going to do this activity to observe the invisible organisms, what we call as microorganisms. So for that, I have a king coconut water that had been kept aside for few days. So from that, what I am going to do is I have a clean glass slide here and a cover slip, and you are familiar with the light microscope. So what I will do is I will take a drop of the coconut water, put it onto the glass slide and then cover it with a clean cover slip. You know how to use the cover slip. You have to hold it slant like this and place it on top so that you get it covered properly. The sample is covered properly. And then we have to make sure there is enough light. So here there is a light source also. Then we place the sample on the stage. And I have to adjust it so that the sample is in line with the light falling on it. Thereafter, under low power, we are going to observe the presence of microorganisms. So now, if I observe it under low power, there you can see the microorganisms clearly. Can you all see the circle-like shaped cells? Those are the invisible organism, what we call as the yeast. You can see it clearly, students. Okay. So that is how microorganism, that is yeast, present in coconut water is visible to you through the light microscope when we observe it under low power. So now we are going to observe some permanent slides that are available in the lab to observe microorganisms. So here I have the permanent slide of a paramecium. This is how you have the permanent slide. So we can use this. This has been already prepared for us. We can use this to observe the different microorganisms. So here we don't have to do anything. We just need to place a slide on the microscope. So whatever the slide that was there, I will remove it. Now I will place the slide on the stage. This is a permanent slide. Then we have to make sure that it is in low power and also that enough light is falling on it. After that, have to make sure the specimen is in line with the light, and then we just need to observe it. Now here you can observe paramecium. Can you all see that shape? A slipper shaped unicellular organism. So it's a microorganism that has a slipper shape. So you can clearly see the shape there. But here we can observe it under medium power as well to get a better observation. So I will change it to medium power. Now if I do an adjustment, you can see it a little bit more clearly. You can see the shape of the cells more clearly. There again you can see the slipper shaped cell more clearly. Can you see it, students? Yes, that's very clearly visible to you. So now I have a amoeba microorganism, a permanent slide of amoeba. So again I will place it under low power. We can observe it here. Can you see the shape? It has kind of an irregular shape because it has the pseudopodia. It has a irregular shape. You can see it clearly. Now I will put it for medium power. Then you will be able to observe it more clearly. Now can you see the irregular shape of amoeba? Yes. So that again is a unicellular organism. Okay, now we will change the slide and I will show you all another microorganism, that is mucor. We have already observed that before. So here again it has to be under low power. Can you all see the shape, students? You can see like a circular shape and thin thread-like structures. That is the mucor. So this is different from the other organisms that you saw. This is a slightly different type of microorganism that you can see. Again, this can be observed through medium power. So if I change it to medium power, can you see it clearly now? Still, you can differentiate it from the other types of microorganisms. So that is mucor, a fungus. So then again, I will use another slide to observe Chlamydomonas. So here, this is a permanent slide of Chlamydomonas. If I adjust it, can you all see the shape? The unicellular organism, like a pear-shaped cell. That is Chlamydomonas. You can see it clearly. Now, if I observe it under medium power, again, you can see it a little bit more clearly. The shape of the cell is more clear to you now. This is Chlamydomonas. Then after Chlamydomonas, what we observed in coconut water, the yeast cell. So that also can be observed using a permanent slide. You know how the yeast cells should look like? Circle-shaped cells. Those are again unicellular organisms. You can see that, observe that clearly. So again, this also can be observed under medium power. So if I set it and adjust it, you can see the shape of the cells, the unicellular organism, even more clearly now. Can you all see it, students? Yes. So now we have observed many different types of unicellular organisms. There is one more, the bacteria. So here again I have the permanent slide of a bacteria that also can be observed under low power. So here if we look at it under low power, can you all see the shape of the cells? This is again different from all the other unicellular organisms that we observe. You can see the bacteria cells clearly. If I put it to medium power again, you will be able to observe it even more clearly. Okay, students. So we observed different permanent slides and you saw they were all microorganisms. So they were unicellular organisms except for the fungus that had a different structure. But you were able to identify the different types of microorganisms. So now we are going to observe a special type of or a certain type of microorganism, that is mucor, a type of fungus that grows on bread. So I have already prepared a slice of bread. I sprayed water and left it aside for 3 days. Now here you can see the growth of the fungus, mucor. So from this we have to take a clean glass slide I have here, and from here I have to take a small sample of the fungus, the piece of bread with the fungal growth, and place it on the glass slide, and then I will have to put a drop of water on it so that you can easily cover it with a cover slip. And here again I have a clean cover slip also. So we place that and now we use it to observe under the light microscope. So I'll remove whatever slide we have to put it to low power, and then here if I make sure that the sample is in place with the light source. So now if we observe it through low power, can you all see the growth of fungus there? You can see that particular type of microorganism grown on bread. Can you all identify the shape of it? Yes. So if we observe this through the microscope, you can confirm that microorganisms, that is a fungus which is called mucor, has grown on bread. The same thing can be observed under medium power also. So if I adjust it there. Now can you all see it more clearly? Can you identify the shape of the microorganism? Yes. So there you saw what mucor is, and you know it grows on bread. So now we are going to observe the microbial activity. To observe that this is the microbial activity on food, the food substance is going to be sugar, and the microorganism that I am going to use is yeast. And in addition to that, we need warm water about 40 degrees Celsius. And here I have 2 beakers into which I am going to take 200 milliliters of water. Before that, into both beakers, I am going to take 2 teaspoonsful of sugar into each beaker. So 2 teaspoons into one and another 2 teaspoons of sugar into another beaker. Then we need to add 200 milliliters of water. This is about 200. It doesn't have to be an exact measurement, but it has to be warm water. Then I will mix it together so that the sugar dissolves completely. We have to make sure that it dissolves completely in both beakers so that we have a proper sugar solution. So now that the sugar has dissolved completely, I will add 1 teaspoon of yeast into both these sugar solutions. We are going to have one mixture like this so that we can observe what happens. Have to mix it properly. And this other mixture we have to transfer into a bottle. Now that it's all mixed, so I can just leave this aside to observe what happens. Here I will transfer this mixture into the bottle. So to this bottle we have to fix a balloon to observe the products. So here we have 2 mixtures, one with sugar and yeast. It's kept open to observe what happens here, and the other one, the same mixture, sugar and yeast, but we fix a balloon to see what will happen. Like this, we have to leave the 2 setup for 20 minutes to get the proper observation. So now I have prepared these 2 types of setup before. I have a bottle where I have fixed the balloon here. You can see how the balloon is inflated. And I have another beaker here. Here also I have already made the same activity. I did the same activity. You can observe here, students. This is how the balloon inflates after about 20 minutes. The reason is when yeast act on sugar solution, there are different products produced. Those are the byproducts. One is carbon dioxide that comes up and fills up the balloon. That's why the balloon gets inflated. Then if we take this solution, here also you can see some forminature. Here you can observe it more clearly. The forminature is there. That is again due to carbon dioxide. If I smell it, now you all can't get the smell, but I can get the smell. It gives an alcohol smell. So that is another byproduct. One is carbon dioxide, the other one is alcohol. And the observations are, you can see the balloon inflating, you can see the form here, that is due to the carbon dioxide produced, and also I can get the smell. So those are the observations when yeast acts on sugar solution.